Hey, this is Dr. T, and I'm continuing my look into metabolism via Wikipedia. Okay, so let's look at protein catabolism. And in this case, we're digesting proteins. So unfortunately, I wish there were some awesome pictures here. This one we are going to look at. But when we're looking at protein catabolism, the first step is to take the protein and turn this into amino acids. Now, if you are consuming the protein for food, so you just had a hamburger, you're going to be processing the carbohydrates from the bun, but you're going to jump into the amino acids and the proteins in the burger itself, then step one is you're going to take those proteins, you're going to turn them into peptides. That takes place in your stomach. The idea here is now it's a lot less dangerous. You've denatured them with your stomach acid just in case it's, something undesirable. Then you break it apart. So now once it gets into your small intestine, those peptides are going to be turned into amino acids. Those are absorbed, shuffled through the bloodstream, and get processed. Now, if you're metabolizing your own proteins, which happens on a regular basis, there's a lot of turnover, you recycle things, then you're going to have enzymes within your cells that are going to turn the protein into amino acids. Now, once you've done this, you've got a couple of options. You can take those amino acids and just use them, flip them right around, and make new proteins out of them. And that's very common. Additionally, you could do some other processing. So let's look at the other processing. Let's say we're going to use it for energy. So the first step is we are going to take the amine and we're going to remove it. So we're going to go ahead, and we got a couple options. This is the urea cycle. And basically what we're doing is we're taking amino acid backbones, which are going to end up being usually acids with some, nice functional groups we can add things to, and we can stick the amine onto those. So we move it from one amino acid to the backbone of another. So we switch what our amino acid is. Now we can do a couple of things. We can be, okay, I'm going to make a backbone. And then I'm going to stick an amine from one amino acid that I don't want onto the backbone of an amino acid that I do want, making the amino acid I want. And we totally do that. Alternatively, animals are typically nitrogen-rich, carbon-poor. So we have to worry about how much carbon we have. But nitrogen, we're literally throwing it out. That's good old number one. So what we're going to do, we got a couple of options to throw away our nitrogen. And it depends on what type of animal you are. For us mammals, what we prefer to do is the urea cycle. So we're going to take our amino acids, we're going to move the amines onto specific amino acids, eventually getting onto arginine, and then we are going to split off part of that and then chuck off the urea, which I'm getting a little bit lost. Here's the urea. So number 7 is urea in the cycle. And then we're going to start it all over again. And, we're not going to go into the aspartate shuttle or anything like that. But just kind of the key take home. You've got this cycle where we've got a couple of amino acids and their backbones. We're shuffling them around, and we are producing this small molecule called urea. We like urea because urea is relatively non-toxic, far less toxic than ammonia. If you happen to be a fish, which I'll be surprised if you are, but if you happen to be a fish, this is kind of like not really needed for fish. I don't know enough about fish biology, let's just be honest. But what fish can do is the ammonia can just go straight out the gills. And so they basically exhale the ammonia, which works fine for fish because they're in an aqueous environment. But for everything else, we have to store it. Birds will go for more of a solid nitrogenous material, which we do as humans produce a bit. This is why bird poop is white. That's the nitrogenous material for the bird poop. But as mammals, we're going to spend most of our nitrogen sent into urea. Urea is low toxicity, not zero, but low. And then it's water soluble, so we just, number 1 to the toilet. And we're done with it. Now, of course, this leaves us, we have the nitrogen dealt with. What about those carbon backbones? That's where the energy is going to be. At this point, we're not getting energy. We're using some energy, actually. So let's talk about those backbones. And amino acids can get broken into kind of 2 categories, kind of 2.5. If the backbone is capable, and sometimes it's the reason you'd be in this kind of both spots is you might very well have two parts of your backbone, so not all of the backbone is going in one spot or another. So you're going to take your backbone, and if you can stick it into a spot that can turn into pyruvate or oxaloacetate, basically anything that's not acetyl-CoA, if you're able to do that, then you can turn it into oxaloacetate, which then goes into phosphoenol pyruvate, which goes through gluconeogenesis. These are called gluconeogenic amino acids, or glucogenic, I should say, amino acids. This is one of the reasons why those ketogenic diets have some interesting quirks to them. You still do produce glucose. Now, if you are running on so low glucose that you can't produce enough for your brain and tissues to function, then your liver will produce ketone bodies. Okay, so if you don't have enough carbohydrates and you can't make enough through gluconeogenesis, you're going to start making a few of these guys, which are called ketone bodies. And these allow organs that cannot run on fat or amino acids directly, like your brain, to still get energy. Now, the downside, of course, of these guys, they can mess with your blood pH. They will give you that fruity scent to your breath, which is known for diabetics, which in the case of severe diabetes, it's not that you don't have enough carbohydrates, you have way more blood sugar than you need, but your body doesn't understand this. That's partially why you have so much, is there's been a breakdown in communications between your islets of Langerhans and your liver. And so your liver's thinking, oh, we don't have any blood glucose. Let me try and up that. Please, anything but up in this case. And we're going to switch over to these ketone bodies. Alternatively, if your diet is so low on carbohydrates that you cannot produce enough blood sugar, then you'll switch over and start using these ketone bodies. These scenarios are not good really in any case. It's going to be hard on your kidneys, even if it's, intentional kind of thing. Yeah, my general thought is on food, just, eat healthier, that kind of stuff. Okay, so back to the amino acids. The ketogenic are ones that are going to turn into acetyl-CoA. Acetyl-CoA are going to go into ketone bodies. And a lot of them kind of can do both, so parts of them are going to go to different spots. But yeah, those will have to go into becoming ketone bodies if you're going to try and process them. And you're not just going to use them for energy. Obviously, if you dump them into acetyl-CoA, you can go through the citric acid cycle, bada boom, bada bang, you get all the energy you need. Yay. So yeah, that's protein degradation and how you use those for fuel. Energy density wise, carbohydrates, proteins all have about the same energy density, roughly speaking. Fats typically have a good deal more. That's partially because they're nice and packed in tighter, and there's more energy per carbon because they're more heavily reduced. So with that said, have a wonderful day.